Hey, hi. So let's welcome to the cDNA. What is the cDNA? cDNA, otherwise we call it as complementary DNA or copy DNA. So what is the cDNA? If you see, the cDNA is nothing but this was the DNA that is cloned from mRNA. So combination of cDNA, which is complementary DNA, inserted into the collection of host cells. So which is together constitutes some partition of transcriptome of an organism. So cDNA is produced from fully transcribed mRNA. So means after post transcriptal modification we produce this whatever the mRNA is produced, from that we will produce the complementary DNA. We call it as copy DNA. So it is found in the nucleus, thereby contains only the expressed gene of an organism. So the normal DNA and complementary DNA or copy DNA is different. The normal DNA will be having non-coding and coding, which means extrons and introns. But when it comes to cDNA, you have only extrons. If you see here, in eukaryotic cells, the mature RNA is already spliced. Hence, the cDNA is produced from mRNA by the enzyme called reverse transcriptase. So they lack introns and readily expressed in the bacterial cell. If you see, while the information of cDNA is powerful and useful as a tool, since the gene products are easily identified, the libraries lack information about enhancers, introns, and other regulatory elements found in the genomic library. So usually you can see the normal DNA which has extrons, introns, all these things. So after transcription, so it also do the same of extrons and introns. So after processing, the introns are removed and only extrons are joined. Here the poly A is the tail part and 7-methyl-guanosine is the cap. So 7-methyl-guanosine is the capping and polyadenylated 200 to 300 A are attached to the tail. So the mature mRNA will go to the cytoplasm. So cDNA is created from mature mRNA from a eukaryotic cell with the use of an enzyme called reverse transcriptase. In eukaryotes, the poly A tail consisting of long sequence of adenine nucleotides distinguishes the mRNA from tRNA and rRNA and can therefore be used as a primer sites for reverse transcriptase. So you can differentiate the mRNA from rRNA and tRNA. They can therefore be used as a primer site. So mRNA is obtained and purified from the rest of RNAs. Several methods will be for purifying the RNA, such as trisol extraction, column purification. The column purification is done by using oligometric T nucleotide coated resins, where only the mRNA having the poly A will bind with that. So oligo DT nucleotide. So the rest of the RNAs are eluted out. The mRNA is eluted by using eluted buffers and some heat to separate the mRNA strands from oligo T. So now we have to inject this mRNAs into bacteria to produce the copy DNAs, complementary DNAs. Later on, RNA-H will be will be transforming the mRNA to DNA. So they will become the double-stranded DNA, which we call it as cDNA. Now this cDNA is introduced into the host cell. So this is one of the technique for introducing the desired fragment of DNA into the host cell. Usually this is used in the case of ADA treatment, adenosine deaminase. Okay, so thank you.